so much. A pleasure to be here and present Cantargia. So obviously, thanks for the introduction, Chris. So Cantargia is very much, as you said, antibodies against one molecular target called IL-1RAP. And IL-1RAP has relevance both in cancer and autoimmune inflammatory diseases. But I will start on the cancer side. So what you're trying to achieve here is that obviously if you have a new molecular target, you have a buffet of opportunities and you have to start somewhere. So we started to look into resistance against different therapies and especially chemotherapies. And the whole idea is to think about a patient having a tumor getting chemotherapy. Obviously you expect the chemotherapy to work initially, but very quickly you will develop resistance against the chemotherapy. So our plan is then to add this antibody, which is then designed to interfere with some of the resistance mechanisms, IL-1, and then you will get a much more durable response and you will get a much deeper response. And the nice thing is that we've done more than 250 patients right now with this antibody. We've done more than 70 patients in pancreatic cancer, which is our lead indication, and we can really see that we get much deeper and durable responses, and this has an impact on prolonged survival, expect to historical data. So we're now having a pipeline where the lead asset nadunolimab is entering a registration trial in pancreatic cancer together with PanCAN in the US. We're in final discussions with the FDA and hope to get that phase 2/3 trial started as soon as possible. We also have nice intriguing data both in non-small cell lung cancer and in triple-negative breast cancer. Lung cancer is still recruiting to get more data in non-squamous non-small cell lung cancer. Triple-negative breast cancer is entering a randomized trial right now as a first/second line opportunity based on, again, promising early data. Second asset, CAN10, is in development for myocarditis and systemic sclerosis, entering phase 1, hopefully during Q2. We are planning to do the submission during Q1, and then obviously we don't really know how long time the regulatory authorities will take here. So starting with nadunolimab then. So as I said, antibody against IL-1RAP, and IL-1RAP is essential for signaling of both IL-1 alpha and IL-1 beta, and this is important. The antibody blocks both IL-1 alpha and IL-1 beta activity by binding to IL-1RAP and blocking the interaction between the IL-1 receptor and IL-1RAP. The antibody also has been designed to have improved ADCC by a defucosylation strategy, so it will be a very active molecule when it comes to NK cell activation and killing of tumor cells in that way, and also very active when it comes to modulating the activity of myeloid cells and fibroblasts in the tumor microenvironment. This is an experiment showing the relevance of IL-1RAP blockade when it comes to combination with chemotherapy. And remember that IL-1 is not one molecule, it's actually two different molecules, alpha and beta. And if you treat cancer cells with, for instance, docetaxel, you will see a very potent increase in IL-1 alpha on the cancer cells. And then if we look at this in vivo, you can see that if you treat a syngeneic mouse here, a mouse model, with docetaxel, you get an anti-tumor effect. If you block only IL-1 beta, there is no synergistic effect, but if you block the IL-1RAP activity, there is certainly synergy here. And this is important because IL-1 beta blockade has been tested clinically and has not shown to have any beneficial effects in combination with chemotherapy. So then moving into pancreatic cancer, I showed a waterfall before. It looks good. We have lots of patients down in this region where you will want them with good tumor shrinkage. This is first-line patients in combination with gem-Abraxane. And what you can also see is that the progression-free survival and the survival is longer than you expect from historical controls. So the PFS is about 7.2 months, about 2 months longer than you would expect from chemo alone. Disease control rate is higher, and the OS is somewhere 3 to 4 months longer than you would expect from chemo alone. So these data were presented at ASCO last year, and we're planning to do an update of the data during second quarter this year. What I should also point out that in addition to response rates of 33%, we have a number of patients that have a very unorthodox delayed benefits of initially progress to then be followed by response. We don't really know the mechanism behind this. It's not the traditional T-cell based infiltration that you see with PD-1s, for instance. Safety profile, very good. There is one thing I'd like to point out first, which is the neutropenia. We do get higher levels of neutropenia, but the good thing is that during this protocol, we started to introduce G-CSF prophylactically, and we see that in the patients that get prophylactic G-CSF, you have a much, much lower incidence of neutropenia. So it's something we can control while we're taking this further into randomized trials. The other part here is super interesting. You expect quite a lot of neuropathy with a drug like Abraxane, and from previous trials, somewhere 15 to 20%, grade 3 or higher. We've only seen 1 out of these 70 patients with serious neuropathy, and that fits very well with the mechanism of action here, which taxane-induced neuropathy is caused by IL-1 and neuroinflammatory responses. There's also some signals here on, for instance, diarrhea and fatigue, but we would like to see randomized data before making any conclusions. In lung cancer, similar story. Here we worked in a mix of first-line and second-line patients that are chemo-naive. Response rate is about twice as high as you would expect from chemo alone. PFS is higher, median survival is longer, and the signals seem to be driven by the non-squamous non-small cell lung cancer population rather than squamous. So we have decided to add more patients here before taking this into a next step development. And last week we finally got our first data in triple-negative breast cancer, and again a very strong activity signal with lots of patients having more than 50% tumor reduction. We have 1 complete responder. We have a 50% response rate in a small number of patients, but still you would expect somewhere around 30%. So if you combine all this information, we have similar types of chemotherapies. We have cancers that are inflammatory and, let's say, have a tumor suppressive inflammatory microenvironment, and we typically see response rates, PFS and OS, which is much better than you expect from chemo. So we are very hopeful that we have something here and are looking forward to taking next steps. So then going into the autoimmune field. So CAN10, which is our second asset, has been designed not only to block IL-1, but IL-1RAP is also mediating signals from IL-33 and IL-36, which are implicated in a large number of diseases. And for instance, in a disease like systemic sclerosis, there are different inflammatory pathways activated by these different cytokines, but we all end up in an uncontrolled inflammation followed by fibrosis and potential death of the patient. So if you look at down in the bottom, then we have generated very nice data which was presented late last year at ACR in various models of systemic sclerosis, and we have a good effect on both skin thickness as well as lung fibrosis, and these are in established models where we see these effects. We also see very good effects in myocarditis models, and myocarditis is a complex disease, but we see if it's virally induced or if it's autoimmune, And interestingly, for instance, here, if you compare the CAN10 result, you can see that it's stronger than if you just do an IL-1, complete IL-1 blockade. So it's definitely an effect by blocking several of these cytokines. And it's similar here in the autoimmune myocarditis. So going forward in the CAN10, we've done everything we should. So the GLP tox studies have been completed at much higher dose levels than we intend in the phase 1. We've not seen anything peculiar or any safety concerns. So basically we're just finalizing the application to be sent off now during Q1, and hope to start study first in healthy volunteers in single dose studies, followed by repeated doses in patients with psoriasis, and we're going to do this as ambitious as we possibly can to get both biopsies from the psoriasis patients as well as blood from both the healthy volunteers as well as the patients to get early proof of concepts that the drug is doing what it's supposed to do. So exciting times here as well. So when it comes to then the news flow, so we have lots of interesting news ahead of us. So you've seen the pancreatic cancer data, the non-small cell lung cancer data, and TNBC data. So for pancreatic cancer data, we expect to give an update during Q2. And we will also include much more on biomarkers. We have collected biopsies from all of the patients, or most of the patients in this trial before therapy, and quite a few on treatment. So we can start seeing what's happening in the tumor and correlate that with long-term treatment effects. Similar story in triple negative, or in non-small cell lung cancer. We're also planning to present more data during Q2 to build in the biomarker. And in triple negative breast cancer, as I said, that the trial was designed as an initial safety lead-in phase. 15 patients, the last 3 had not been treated long enough to have any CT scans, but the 12 looks very good. We will present data second half of the 15 patients with more long-term follow-up. We also have, since we have a randomized trial, we have a first interim analysis during Q4, which will be some kind of futility analysis, and obviously we will keep you updated of what's happening in that one. And then we have done a trial which I haven't presented here on the Keytruda combination, and that was also presented at ASCO last year, with small trial with some patients doing very well, and we'll present more data on that as well once it's mature enough. Then we have the phase 2/3 trial, which is upcoming and starting. As I said, it's done in collaboration with PanCan in the US. It's a phase 2/3 trial, and we hope to submit the protocol in the near future. Preclinical translational results, yes, more. And then we have a number of other trials where we're not recruiting. We stopped recruitment during the autumn, but we are now getting more data on the FOLFIRINOX combination in pancreatic cancer, again, first line. We did a basket trial looking into non-small cell lung cancer, docetaxel combination, colorectal cancer, FOLFOX, third line, and biliary tract cancer. And all those data are now maturing, so probably H2 would be a good time point to present those. And the CAN10 program, also good news flow during this year. So then to end up a little bit here, we have a solid cash position. We had about 427 million Swedish, which is about 41 million US on the bank end of Q4. It will take us until about mid-2024. The company is listed on NASDAQ Stockholm. We have a market cap of around 110 million US, or 1.2 billion Swedish. And we have very strong owners, so we have several pension funds, bank funds, and a few specialist investors further down the list supporting us. So to sum up, Cantargia is very much focused on new antibodies against IL-1RAP. where we have a potential to address several severe diseases, both within cancer and autoimmune diseases. We are focusing on pancreatic cancer, triple-negative breast cancer, non-small cell lung cancer right now in the cancer, and that keeps us very busy, as well as getting CAN10 into a clinic. And we have, and I see that the cash position has not been updated, so excuse me for that. But we have a cash runway until mid-2024. And to finish off with, we have a very strong patent portfolio. We have obviously composition of matter patents on our antibodies until 2035 for lead compound and 2041 for CAN10. But we also have a robust patent portfolio covering antibodies for treatment of cancer in more general, which is valid until 2032. So by that, I'd like to thank you for attention and very happy to take some questions.